Carbohydrate metabolism involves several processes. Cellular respiration is the process by which your cells synthesize ATP. The main fuel used for cellular respiration is glucose. When blood glucose levels become elevated, the glucose can be stored in the form of glycogen through a process called glycogenesis. When blood glucose levels begin to drop, the glycogen that has been stored will be broken back down into glucose through glycogenolysis. New glucose molecules can be synthesized from non-carbohydrates, such as proteins and fats. So what is ATP? You can't live without it, and it is the energy currency for all living organisms. ATP, remember, stands for adenosine triphosphate. The tri refers to the fact that there are three phosphate groups bound to a ribose and an adenosine molecule. The chemical bonds between the phosphate groups are high energy bonds, so when you break one of those bonds, such as taking ATP and breaking it down into ADP plus phosphate, the energy in that chemical bond then can be used to carry out cellular work. Most of the ATP synthesized by your cells uses aerobic cellular respiration. Aerobic refers to oxygen. This type of cellular respiration requires oxygen. This oxygen is coming from the air that you breathe, being transported by your hemoglobin to the tissues. Let's look at the processes. Aerobic cellular respiration involves the oxidation of your food fuel. Glucose will be the main food fuel. This reaction demonstrates aerobic cellular respiration. The C6H12O6 is glucose. Glucose reacts with oxygen to form carbon dioxide and water. The carbon dioxide and water are products of aerobic cellular respiration. In addition, energy will be released to synthesize ATP. ATP can be synthesized by substrate level phosphorylation, which involves a chemical reaction where phosphate from a substrate is removed and attached to or binds with ADP to form ATP. Oxidative phosphorylation is more complicated, and it involves an electron transport chain. Three pathways are involved in aerobic cellular respiration: glycolysis, the citric acid cycle, and oxidative phosphorylation. Well, let's get started with the oxidation of glucose. Remember that glucose in the presence of oxygen is going to be broken down into water and carbon dioxide. The energy in the chemical bonds of the glucose can be used to make about 32 ATP per glucose molecule. In addition, some of that energy is going to be lost in the form of heat, and we're going to use this to keep our bodies warm. The first pathway in the oxidation of glucose is glycolysis. And glycolysis occurs in the cytosol of the cell. Remember, the cytosol is the fluid portion of the cytoplasm, which is located between the nucleus and the plasma membrane or the cell membrane. Glycolysis can occur in the absence or the presence of oxygen. It doesn't make any difference, and so it doesn't require oxygen, but it certainly can occur when oxygen is present. You're going to begin with one glucose molecule, and it will be broken down into the end products of two pyruvic acid molecules. And in the process of glycolysis, you are going to reduce two NADs into NADH plus H. So remember, reduction is the gaining of electrons, and we can see those electrons in the hydrogen ions here. The NADHs will be transporting those electrons to the electron transport chain. So NAD, recall, is a coenzyme and its job is to transport electrons. The energy in the glucose chemical bonds can be used to synthesize 4 ATP molecules. However, you're going to use 2 of those, so that leaves you with 2 that you can use for cellular work. And so that's going to be a net gain of 2 ATP per glucose molecule. The ATP are synthesized by substrate level phosphorylation. This is an illustration of glycolysis. It involves 10 steps, and each step is going to require a specific enzyme for it. You're not going to have to learn each one of these steps, but I'm going to show you what I do want you to know, and we're going to break it down and kind of follow through this. If you look at the very beginning, up at the top, you have glucose, and then if you'll follow down to the bottom, you'll see that you have 2 pyruvic acid molecules. Let's take a closer look at glycolysis. Recall, glycolysis occurs in the cytosol of the cell. It can take place in the presence of oxygen or in the absence of oxygen. It begins with glucose, a 6-carbon molecule. The gray balls represent carbon. During the first reaction, ATP is broken down into ADP and phosphate. The phosphate binds to the glucose, forming glucose 6-phosphate. The number 6 represents the carbon to which the phosphate is attached. It is attached to the 6th carbon. The carbons then are rearranged to form fructose 6-phosphate, another monosaccharide. A second ATP comes in and is broken down into ADP and phosphate, and its phosphate binds to the first carbon to form fructose 1,6-bisphosphate. Bisphosphate just means 2 phosphates. This 6-carbon molecule then is split into 2 3-carbon molecules called glyceraldehyde 3-phosphate. At this point, the pathway has branched. In the previous slide, I described how fructose 1,6-bisphosphate is split into 2 3-carbon molecules called glyceraldehyde 3-phosphate, or G3P. At that point, remember, the pathway branched. This slide shows you the 2 branches of the pathway. So we're going to follow the reactions that occur in these branches. They're going to be identical in both branches. So let's begin with the G3P. Notice that a phosphate comes in and will bind to the G3P. At the same time, NAD, a coenzyme that is used to transfer electrons, will come in and remove a pair of electrons from the G3P. When NAD picks up those electrons, it is reduced. Recall the saying, Leo goes Gur. Gain electrons, reduction occurs. And we can follow those electrons by following the hydrogen. So you can see that NAD becomes NADH. The new molecule then that has the 2 phosphates attached to it and the pair of electrons that have been removed is called 1,3-bisphosphoglycerate, or 1,3-BPG. In the next step, ADP comes in and removes one of those phosphates from the 1,3-BPG. It's going to remove the first one to form a molecule called 3-phosphoglycerate, or 3P. And in the process of doing this, ATP is being synthesized. So at this point in the pathway, we have synthesized 2 ATP molecules because we have 2 branches. You then have a rearrangement that occurs. You're going to be moving the phosphate to a different carbon, and we're going to be going through several steps here, changing the 3P into the 2-phosphoglycerate, or 2P, into the phosphoenolpyruvate, or the PEP. Once we get to that point, another ADP comes in, removes the phosphate of that 3-carbon molecule to form ATP. Again, since we have 2 branches, we're going to have 2 more ATP synthesized, and the remaining 3-carbon molecule is the pyruvic acid. So for each glucose molecule, you are producing 2 pyruvic acids. We used 2 ATP at the very beginning of the pathway, if you refer back to the previous slide, but we've also synthesized 4 ATP. So we made 4 ATP by substrate level phosphorylation, used 2 of them, and that leaves us with 2 for cellular work. And so that's a net gain of 2 ATP per glucose molecule. These ATP were synthesized by substrate level phosphorylation. The pyruvic acids that remain have most of their energy still within the chemical bonds, so they've only released a small amount of energy, just enough to make the ATP through substrate level phosphorylation. The electrons that were picked up by the NAD are going to be transferred to the electron transport system, and those electrons will be used to make additional ATP through oxidative phosphorylation. As a matter of fact, most of the ATP synthesized during aerobic cellular respiration will be synthesized at the electron transport chain through oxidative phosphorylation. I included this slide because I wanted to show you what happens to 1,3-BPG, or the 1,3-bisphosphoglycerate. I've got a circle around the reaction where 1,3-BPG is converted into 2,3-BPG, or 2,3-bisphosphoglycerate. That should ring a bell. Do you remember that 2,3-BPG, as those levels increase, that stimulates the unloading of the oxygen from the hemoglobin? And so we can see that cells that are metabolically active can produce 2,3-BPG, and that can stimulate hemoglobin to unload its oxygen. If oxygen is not present, anaerobic cellular respiration occurs. This is called fermentation. In this slide, you'll see pyruvic acid on the left. That was the end product of glycolysis. Recall, we had 2 of these, so this reaction will happen twice. Pyruvic acid in mammals will be converted into lactic acid, and NADH will be converted back into NAD. So NADH has to be oxidized back into NAD so that it can return back to glycolysis. Without doing this, glycolysis stops. The purpose of converting pyruvic acid into lactic acid is simply to oxidize the NADH back into NADs to return back to glycolysis. You can follow those hydrogens that are on the NADH, and you can see where the hydrogens have been bound to the pyruvic acid, converting it back into lactic acid. Notice no ATP are synthesized in this last step. So during anaerobic cellular respiration or fermentation, you only have a net gain of 2 ATP synthesized by substrate level phosphorylation. And these are the 2 net gain that you saw in glycolysis, being produced during glycolysis. If oxygen is present, and only if oxygen is present, will the 2 pyruvic acids formed during glycolysis enter the mitochondrium. Cellular respiration that occurs in the presence of oxygen is called aerobic cellular respiration. That is why you breathe in air, so that you can undergo aerobic cellular respiration. What we're going to see is that most of the ATP synthesized per glucose molecule will be synthesized during aerobic cellular respiration. In the presence of oxygen, the pyruvic acids will move into the mitochondrium. The mitochondrium is an organelle called the powerhouse of the cell. It's called the powerhouse of the cell because this is where aerobic cellular respiration occurs. It consists of two membranes, an outer membrane and an inner membrane. The space in between the two membranes is called the intermembrane space. The inner membrane is highly folded to increase the surface area, and these folds are called cristae. The inner membrane has molecules that make up the electron transport chain embedded in it, and the electron transport chain is involved in oxidative phosphorylation. Within the inner membrane is a matrix. The matrix contains the enzymes involved in the citric acid cycle, the next step of aerobic cellular respiration. Let's move on to the next step in aerobic cellular respiration, the citric acid cycle. The citric acid cycle occurs within the matrix of the mitochondrium. That's because the enzymes for the reactions for this cycle are within the matrix. Remember, this requires oxygen. There are two phases. In the following slides, I'll talk about these phases, but they involve the transitional phase and the citric acid cycle. Let's look at the transitional phase. The pyruvic acid moves into the matrix of the mitochondrium. Once it's in the matrix of the mitochondrium, it will lose a carbon in the form of carbon dioxide to become a 2-carbon molecule called acetate. NAD then comes in and picks up a pair of electrons, and we can follow those electrons with the hydrogens to become reduced to NADH. The NADH then will transport the electrons to the electron transport chain. Also, we have a coenzyme A that comes in and picks up that 2-carbon molecule, the acetate, to become acetyl-CoA. The acetyl-CoA then moves to the citric acid cycle, and we'll pick up with that once we get to the citric acid cycle. So let's recap what is occurring per pyruvic acid molecule. Remember, 2 pyruvic acid molecules are being formed during glycolysis, so everything that we have per pyruvic acid molecule, you must multiply times 2 if I'm asking for you to tell me what is being produced per glucose. So per pyruvic, you have 1 carbon dioxide that's being formed. This is the first time carbon dioxide is produced as a waste product of aerobic cellular respiration. You have an NAD that was reduced to NADH, and you have an acetyl-CoA. If I were to ask you per glucose molecule how many are being produced, you would tell me 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoAs. The acetyl-CoA that was formed during the transitional phase will move to the citric acid cycle within the mitochondrial matrix. The CoA will then unload its cargo, the 2-carbon acetate, and return back to the transitional phase to pick up a new molecule of acetate. The acetate will bind to oxaloacetate, which is the 4-carbon molecule. Carbohydrates are required to synthesize oxaloacetate. In the absence of carbohydrates, the citric acid cycle will stop due to the fact that you can't make oxaloacetate. When the oxaloacetate and the acetate combine, they form a 6-carbon molecule called citrate or citric acid. Each pyruvic acid spins the citric acid cycle 1 time. So let's look at the products produced during 1 citric acid cycle. 3 NADs are reduced to NADH per pyruvic acid, and 1 FAD is reduced to FADH. FAD is also a coenzyme, flavin adenine dinucleotide. Both NADH and FADH carry their high-energy electrons to the electron transport system, where they will be used to synthesize additional ATP. You also produce 2 carbon dioxides per pyruvic acid, as well as 1 ATP by substrate level phosphorylation. Because glycolysis begins with 1 glucose molecule and ends with 2 pyruvic acid molecules, each glucose molecule then will be responsible for producing 6 NADHs, 2 FADHs, 4 carbon dioxides, and 2 ATP. This is because the citric acid cycle must occur twice. Let's do an energy count so far. Remember, ATP is your energy currency, and NADH as well as FADH are transporting high energy electrons to the electron transport chain, so they are a source of energy as well. Let's do the count per glucose molecule. Recall, during glycolysis, 4 ATP were synthesized, but 2 were used, and so you have a net gain of 2 ATP through substrate phosphorylation, and this is available for cellular work. In addition, 2 NADHs were formed, and those NADHs are transferring their electrons to the electron transport chain if oxygen is present. During the citric acid cycle, the first portion of it, the transitional phase, 2 NADHs are being produced per glucose molecule. Remember, it was 1 NADH per pyruvic acid. No ATP are produced during the transitional phase. During the citric acid cycle, 2 ATP are produced per glucose molecule by substrate level phosphorylation. 6 NADHs were produced, so you take the 3 that were produced during one cycle of the citric acid cycle per pyruvic acid, and since we have 2 of them, we're just multiplying everything times 2, and 2 FADHs. So up to this point, the net gain so far has been 4 ATP that have been produced by substrate level phosphorylation, 10 NADHs, and 2 FADHs. Remember, the NADHs and the FADHs will transport their electrons to the electron transport chain to make additional ATP. Now let's look at the carbon dioxide in the water. Per glucose molecule, during glycolysis, no carbon dioxide or water was produced. During the citric acid cycle, in the transitional phase, 2 molecules of carbon dioxide were produced per glucose, but no water was produced. During the citric acid cycle, per glucose molecule, 4 carbon dioxides were produced, but no water. Remember that the equation for aerobic cellular respiration involves glucose plus oxygen on the left side of the arrow being converted into water and carbon dioxide. So at this point we haven't produced any water. We're going to see how the water is produced when we get into oxidative phosphorylation. The third phase of aerobic cellular respiration is oxidative phosphorylation. Oxidative phosphorylation involves the electron transport chain and a process called chemiosmosis. We'll look at each one of these processes. The electron transport chain is located within the inner mitochondrial membrane. Before moving on, let's review redox reactions. Remember, Leo goes Ger. Lose electrons, oxidation occurs. Gain electrons, reduction occurs. Recall, We can follow those electrons by following hydrogen ions. Hydrogen ions are also protons. The electron transport chain consists of enzyme complexes numbered 1 through 4. Each complex is involved in accepting and donating electrons. When a complex accepts or gains an electron, it is reduced. When it donates or loses an electron, it is oxidized. Therefore, these complexes are involved in redox reactions. The electrons are pulled through the chain as they move toward oxygen. They are attracted to the oxygen, and the oxygen is therefore the terminal electron acceptor. Complexes 1, 3, and 4 also function as proton pumps. Remember, hydrogen ions are also protons. These proton pumps pump hydrogen ions from the matrix into the intermembrane space, creating a hydrogen ion or proton gradient. This gradient will be used in the next process called chemiosmosis. The second phase of oxidative phosphorylation is called chemiosmosis. During this phase, the protons that are in the inner membrane space move back across the inner membrane into the matrix. Since the inner membrane is impermeable to the protons except for at proton channels, the protons then move through the proton channels. These channels also function as an enzyme, ATPase synthase. As the protons are moving along their concentration gradient from an area of high proton concentration to an area of low proton concentration, energy is released. This energy that is released is used to synthesize ATP. Think about a ball rolling down a hill, going from an area that's high to an area of low. That ball, as it rolls down the hill, releases kinetic energy. It's the same concept here. Let's recap. Oxidative phosphorylation. The electron carriers NADH and FADH carry their electrons to the electron transport chain. Through a series of redox reactions, the electrons release their energy one step at a time, and that energy will be used to pump protons from the matrix into the inner membrane space. You have to pump the protons because you are pumping from an area of low concentration, the matrix of the hydrogen protons, into an area of high concentration. It's like pushing the ball up the hill. This creates a proton gradient. During chemiosmosis, the proton gradient will be used to synthesize ATP. The protons that are now within the inner membrane space will come back through the membrane into the matrix through proton channels, which are also referred to as complex 5. Recall, these channels function as the enzyme ATP synthase. Synthase means to make, and so this enzyme is involved in the reaction where ADP binds with phosphate to create ATP. The protons that were pumped into the inner membrane space release the energy as they flow through this proton channel or complex 5, and that energy will be used to synthesize ATP. Oxygen is the terminal electron acceptor as well as the acceptor for these protons. When the electrons and the protons bind to the oxygen, water is formed. Let's look at glycogenesis and glycogenolysis. Glycogenesis is taking the excess glucose and converting it or storing it in the form of glycogen. The hormone involved in stimulating this reaction is insulin. Insulin is released while you're eating. Glucose that is not immediately used for energy to produce ATP is stored in the form of glycogen. The organ where glycogenesis occurs is primarily the liver. The beta cells of the pancreas are the cells that release the insulin. Glycogenolysis is taking the glycogen and breaking it back down into the glucose. This occurs when your blood glucose levels begin to drop in between your meals, and the hormone responsible for this reaction is glucagon. This reaction will occur primarily in the liver as well.